Hey, everybody. So I'm going to give a brief lecture on sequence capture and reduced representation sequencing. So you may have heard of like RAD sequencing or GBS, that's genotyping by sequencing. These are all examples of reduced representation sequencing. Okay, so first we'll talk about sequence capture. So the first thing to talk about is experimental design, right? So you'll see the term targeted sequence capture. You also see the term HiBSeq. They're kind of used interchangeably. This is a powerful method that's basically transformed phylogenomics and population genomics. So it's useful in both of those areas. So there are lots of different implementations of sequence capture. It's not just used in phylogenomics, it's used in like cancer research and things all over the place. So detecting COVID-19, etc. Okay, so there are lots of different implementations. So for example, targeted sequencing of ultra-conserved elements. These are parts of the genome that are very, very well conserved between broad phylogenetic distances, allowing them to be used to capture those regions usually for larger scale phylogenomics. Although they tend to be useful also down to the species level, basically. There are a number of pre-designed sequence capture probe kits that are available for ordering. One of the more recent and useful ones is the Angiosperms 353. Okay, that's going to basically sequence up to 350 nuclear loci across the genome for angiosperms. And then there are some for more specific orders or families. For example, there's one for the Compositea, the Asteraceae. There's one for palms that was used very recently. It captures like 4,000 loci across all palms. You can also design your own probe kit based on information from transcriptomes, closely related genomes that might be available. So we did this in sable palms, Hayduk et al. 2016. We did it across the Zingiberales, which is like 100 million years of divergence. Okay, so if you have sequenced genomes, that's a really useful tool. But you can also do this from transcriptome sequences, or even from genomic or transcriptomic resequencing studies. So if you have multiple individual sequences, you can use that to detect polymorphic regions that are conserved and orthologous between your samples, between those samples to generate probes. Or you could also just build your own probes in the lab. So everything I've talked about before this requires another outside organization to actually synthesize the probes for you. So you would design informatically your own probes based on information you have, and then you'd go to somebody like Arbor Biosciences, for example, who could then synthesize the probes for you. But you could also build your own probes in the lab. And so there are lots of new methods coming out that we're going to talk about here that use techniques like RAD sequencing and tagging with biotin to actually synthesize, well, not synthesize, but generate your own probes. And then you can use those for sequence capture for a larger set of samples. Okay, so some key readings on sequence capture are listed here. We're going to cover two of those in our boot camp. That's McCain et al. 2018 and then Anderman et al. 2020. So those are both really good resources. But there's been over the last 10 or so years a number of papers written that could be very useful for you. Okay, so if you do decide to design your own probe kit to be synthesized, again, you'll need some kind of information from a genome or transcriptomes or even resequencing data. Okay. Luckily, there are a number of pipelines that allow you to use this information to design the probes, right? So there's, for example, Marker Miner is a good one, and these others that have been sort of dedicated to designing sequence capture probes, mostly for phylogenomics. But I'm going to show you in a minute that there are some added bonuses. Even if you're going after phylogenomic information, say like at the species level, you're going to get a lot more information than you initially intended. Okay, so just an example here, we did this back in 2016 for the Zingiberales. So that's the order that includes the banana and ginger families, costus, birds of paradise, etc. So we had access to 7 transcriptomes from the 1KP project. So basically one for each of the 8 families of the Zingiberales. And we also had the banana genome, which had just recently been published. And so we were able to use those transcriptomes to align those to basically the exons from the banana genome. And then we were able to apply various filters to those mappings and come up with a common set of specific exon baits that we used for sequence capture across a larger sampling of the Zingiberales. So multiple individuals from each family, or multiple species from each family. And we ended up getting like 300 or something, or 500 loci, and we were able to sequence those. Okay, so back to the Anderman paper, there are 4 basic research questions that you need to address before you start a sequence capture project. So first is, what is the intended phylogenetic or population genomic scope of the project? So that will determine your approach in terms of going after certain loci. Do you need more conserved loci across greater phylogenetic distances, or are you looking for high variation regions like introns or intragenic spacers that will be useful at the population level? Is there a pre-designed bait kit already available that you could use that satisfies the requirement of your research questions? What kind of tissue are you working with? So are you working with fresh tissue collected from the field, silica dried tissue, or historical samples from herbarium specimens? Based on the way we do library preps, this kind of doesn't matter. There are some things you have to take into consideration, like degradation, et cetera. But honestly, sequence capture is really powerful and is kind of, in a way, rekindled this sort of herbariomics field. So using herbarium or historical samples to fill sequencing gaps, or in some cases even to study very rare or extinct species that are no longer available as fresh tissue. This is also used very commonly for ancient DNA studies. So sequence capture, if you have ancient DNA that's at very low concentration, it's very degraded, you can still make libraries from it and capture those parts of the genome. And lastly, how can you optimize costs by pooling samples together to do the captures using available sequence data for bait design while ensuring sufficient coverage of all targeted loci? So we'll talk about pooling in a little bit here. Okay, so this is how sequence capture basically works. So first you have your probes. So these are designed specifically to certain parts of the genome that you're targeting. So you can design new probes like we talked about before, or use a pre-designed probe kit. And typically what probes are they cover your entire region of interest across the genome. So if those are exons or a specific set of single copy or low copy nuclear loci, typically these are synthesized as 120 base probes. So these are oligos basically that are typically they're RNA probes, because the RNA is obviously a much stickier has a much stickier properties than double-stranded DNA that you have to denature. But so these are 120 base segments that overlap in a certain way. So your 120 base probes will overlap at 60 bases. And usually what you do is get what's called 2x tiling. So each probe is represented by each part of the target region is represented by two probes essentially. So it's kind of like bricks or something, right? You're tiling at 2x density across the targeted regions. Okay, so you have your probes, and you have your genomic DNAs. So you've prepped your libraries, you've barcoded them, usually forward and reverse dual indexing, so you can maximize the number of samples you barcode. And then what most people do is they will actually pool their samples or pool their libraries at something like 4x or 8x, 16, up to 24, even higher, 48 in some cases, depending on the number of samples you have, the number of loci times locus length, your target coverage depth, and of course genome size and ploidy are all considerations you'd make here. But most people tend to pool their genomic DNA libraries because you can actually get more distance out of your probe kit that you've spent a few $1,000s on or developed yourself or whatever. So you can cut costs by pooling your libraries before you do the sequence capture. Okay, so let's say you've pooled your libraries at 8x and you're doing 8 captures for 64 samples. All right, so you then hybridize your libraries to these usually pre-designed probes. Okay, so you denature your library and then you bind your baits to the libraries. We also include what are called blockers. So if there are any regions of low complexity like microsatellites or other kinds of repeat regions, these can actually dominate your sequence capture by capturing like all the low complexity regions of the genome. So you want to try to minimize the effect of that. So you often add certain oligo sequences that are low complexity that will actually bind to those regions in your libraries and block them from the probes. Okay, so the next step is to bind your probes in your libraries, which are hybridized, to magnetic beads. And this is usually done by a biotin molecule and a streptavidin molecule that are on the beads. Okay, so then you have magnetic beads and your probes and your libraries bound together. You use a magnet to separate them out, remove the supernatant, wash it a few times, and basically what you're doing is washing away all the parts of the genome that are not in your target region. And then you would take them off the beads, amplify them based on the adapter sequences on your libraries, and then you'd send them off for sequencing. And it's good to think about coverage when you're doing a project like this. So again, how many samples, how many target regions, what is the average size of those target regions, and how many reads do you need, really? That's the important question. How many reads do you need to get a certain X coverage? And something like 20X coverage is sufficient for most projects. So that's going to allow you to do things like build complete contigs in phylogenomics or even call SNPs if you're talking about population genomics. So back to the coverage issue. So basically there are 3 things you should consider here. G, the genome size, and in this sense we don't actually mean the genome size, we mean the total length of all the target regions that are covered by your probes. So if you're targeting 2 million base, or 2 megabases of total sequence out of a genome that's a gigabase, that's a small fraction of the genome, but you want to use that number for your target genome size. Read length is another important one. So for paired ends, this usually is like 2 by 100, 2 by 150, or if you're using say like the Illumina MySeq, that could be 2 by 250. So this goes into your coverage calculations. And of course the number of reads. So usually this is the thing you want to know. So you have to solve for n here. So given that you want, say, 20x coverage of your target region, how many reads do you need to get there, given your read lengths and the total size of the targeted region? And just below, I put some typical prices and how many clusters you get. That roughly translates into the number of reads you get. So unique clusters here. So on the Illumina MySeq, you could go 10 million clusters. In many cases, that's enough to do a sequence capture project. Let's say if you have 50 or 60 samples, you're targeting 20x, 2 million bases of the genome. That might be enough to get you to your target coverage. If you're doing a larger project, like sampling more of the genome, so if you want like dense SNP information across many individuals of the same species, or if you're doing a larger scale phylogenomic study with hundreds or even 1,000 taxa, you're probably going to need more sequence depth, and in that case you'd go for something like Illumina HiSeq, or we have an Illumina NextSeq 2000, which can give you up to 2 billion reads per run, or there's NovaSeq, which is even higher, I think, in some cases. But in this case, you're going to need more coverage to get to that target mean coverage across the genome. So just to show you some kind of pricing options here, and these are internal prices at WVU and our shared sequencing facility at Marshall University. So just to give you an idea of like, you know, the cost, the amount of data you need to actually meet your goals with your project. So, okay, so you've sequenced your data, you get your data back, now what do you do? Luckily, there are a number of dedicated pipelines that are specifically designed to deal with sequence capture data. And usually what you would do is you would just use the sequences from your probes. That's usually just a big FASTA file. So the sequences from your probes or the genes that were used to design them from, including the exons and maybe even protein sequences or translated protein sequences. And you'd use those to map your reads back to back to those probes. So you could either do a de novo assembly of your probes and then map those back, which I think is what Piper does. This one is very commonly used in these kinds of pipelines. More recently is CCapper, which has lots and lots of different capabilities like allele phasing, extracting SNPs, etc. And so you can choose based on your project goals which of these pipelines is the most appropriate. And so what you would end up getting is a number of sequences, right? So like FASTA files or something that you'll have to then align and take to downstream analyses, depending on what your project is. One of the added bonuses of sequence capture, so usually sequence capture probes are designed for more conserved regions like exons, but you get an added bonus, and that's called the splash zone. So if you look at the figure on the right here, usually we would tile our probes across the exon sequences. Say we're using a transcriptome to design our capture probes, but we'd basically have like messenger RNA sequences, which are usually concatenated exons or spliced exons, and so our probes would be designed based on those exons, but really you're capturing genomic DNA. So whatever of your libraries match those tiled probes, you're going to not only get exon sequence, but you're also going to get this kind of region off the edge here into intron sequence and also intergenic spacers, et cetera. And those can be really useful for population genomics because they're typically they typically have higher levels of polymorphism. They're not under the same selective pressures. They're not as evolutionarily constrained as coding sequence like exons, for example. And so what you end up getting is really high coverage of your exons based on the probes you designed. And then you get what's called the splash zone off the edges. So you start to get, you get lower coverage, but you extend your sequence capture out beyond those exons into intron and intergenic spacer regions. And if you're interested in high variation markers for, say, phylogeography, population genetics, et cetera, you can use those markers for those kinds of studies. So it's kind of an added bonus here. And the longer your library, the longer insert sizes you have for your library, so the larger your fragments, the more of those regions you're going to get. So say one end of your library fragment matches part of the probe here, you're going to capture that all the way out to the end of that fragment. So a nice little added bonus for population genomics here. Okay, so what are some downstream applications? So now you've captured your data, you've run it through one of these pipelines and filtered your data, et cetera. So if you're interested in phylogenomics, typically what you do is you'd align sequences for each captured locus. So if you captured 500 loci, you do 500 alignments, and you probably use a script to automate that process. You don't, because otherwise it's a lot of clicking. So you can use a script to align all of your loci. And then you build gene trees. So you could use something like RAXML or IQ-tree if you want to use maximum likelihood, Mr. Bayes, et cetera. If you want to use a Bayesian method, there are lots and lots of different ways to build trees. So it turns out like with sequence capture, building your gene trees and getting good gene trees is really important, right? And then once you had your set of, say, 500 gene trees, you'd use some kind of coalescent method to summarize those gene trees and get an estimate of your species tree. So a very common one is Astral in its various derivatives. Here are two of the citations for Astral. You could use something like SVD quartets, which can be used on a concatenated alignment, for example, if you had gene information. So it's a different approach to coalescent phylogenetics. So like I said earlier, the informativeness of your gene trees is key. So if you have a lot of like really small regions that aren't specifically informative, and you have a lot of unresolved gene trees, you're going to have an unresolved or lowly, weakly supported coalescent species tree, right? So the old quote, garbage in, garbage out applies here, right? So highly informative gene trees are key to getting a highly informed species tree estimate. Okay, so you could also just filter your data set to analyze SNPs only. And so what I mean here is with these coalescent methods, you actually have multiple sequence alignments for each one of your loci. Those are going to include variable sites and invariant sites. So they're just going to be the actual sequences that you captured. What we do with a SNP data set is, and this is also used in population genomics frequently, if we call SNPs, then our matrix is just going to be the variable sites. Well, it turns out that modeling SNPs only is pretty difficult. So we have to do some certain types of corrections or take different approaches to analyzing those kinds of data sets. And you could use software like Snap, which is implemented in the BEAST package, or BEAST 2. You could also use something like SVD Quartets, which is based on site frequencies and probabilities. So you have to use different types of methods when you're analyzing SNP data sets only in a phylogenetic context. And I would say the methods are a little behind the technology here. So look for new methods coming out for analyzing SNP data sets. Okay, the other thing you could do is concatenate your loci. So you could concatenate them, and then you could use data model partitions. So like by loci, by codons, et cetera, if you're only analyzing exons. You could do this with partition finder or model finder, for example. And then you could use something like RAXML, IQ tree, or a Bayesian method to build your trees. However, remember, if you're just concatenating your loci, you're treating that entire alignment like a supergene with one phylogenetic history. And what we know from the last 15 years of coalescent theory is that can be statistically not insignificant, but inconsistent. So you could potentially get high support for the wrong answer, especially if you have a lot of something like incomplete lineage sorting, very short, deep internal branches. If you have evidence of hybridization, so a lot of us are working on polyploids, like allopolyploids, like of course that's going to be an issue, right? Also lateral gene transfer, all these things that can confuse phylogenetics programs can be exacerbated by concatenation. So in some cases, concatenation will give you the same answer as coalescent methods. In other cases, it could give you high support for the wrong answer, or it could be statistically misleading in some cases. Okay, so if you're interested in population genomics questions, typically the path you would follow would be to map your reads to some kind of reference, whether that's your, if you've done sequence capture, you can map them back to your probes. If you don't have a reference genome, you could map them to a closely related genome, not the best thing to do. If you have a genome of the same species, that would be the ideal thing to do here. You could do a de novo assembly of your captured reads and then map to that as a pseudo reference. So typically you map reads with like a fast read mapper like BWA or Bowtie or something like BBMap. And then you would filter and sort your mappings with things like SAM tools and Picard tools. And you could do this based on mapping quality. You could remove PCR duplicates, which can give you inflated estimates of coverage, et cetera. And then once you've filtered and sorted your mapped reads, you can then call SNPs. Okay, so GATK is a really powerful one. Freebase, there are lots of examples of SNP callers, but a lot of people are using GATK because it'll call SNPs, then it'll realign your reads, and then call SNPs across your whole data set. So it's really powerful in that respect. And apparently it can call reads for polyploids now as well. So that's an added advantage. So usually we assume SNPs are diploid or biallelic, but this can actually account for polyploid samples. Okay, then you could do some additional filtering based on quality. If you're only interested in biallelic SNPs, then you could filter based on these parameters. You can filter for presence across samples. So like, for example, if you want to be really conservative, you could say, well, I'm only going to call a SNP if it's present in 90% of my samples. And so really what you're doing is you're taking this big data set and making it smaller based on more stringent filters, but that's good practice. You could filter on minor allele frequencies. So like, you could set this to like 90% or conversely 0.1. So anything under a frequency of 10% or something gets removed. This is pretty common in population genomics. And then downstream, once you have your SNP data set, you can do things like FST estimation, look at population structure, inbreeding coefficients, heterozygosity, and other kind of more basic population genomics or population genetic metrics. You could do analysis of molecular variants to look at the different strata of divergence among your subpopulations, sampling localities, and then of course among and within individuals. You can estimate rates of gene flow based on these data. You can do structure analyses, use either structure, admixture. This one is becoming really popular. That's discriminant analysis of principal components. It's also really fast and shown to be just as accurate as structure. You could do scans for FST outliers, which might suggest local adaptation. And then you could also build population trees or networks using something like SVD quartets or TreeMix. Okay, so just some examples of what your output might look like if you're doing phylogenomics. We're used to seeing like bootstrap support values along branches, but once you're start dealing with 500 or 1,000 loci, now we're looking at pie charts, and these are basically what they're showing you are the number of loci supporting a specific set of relationships. So this example with Apis next to Bombus, obviously these are bees here, honeybees, bumblebees, like 3/4 of all the loci suggest this relationship. And then it also gives you an idea of conflicting relationships. So what percentage of loci that you included support some other set of relationships, okay? So these are mostly based on quartets. And I think 5 parts is the program that's run with Astral here to give you this kind of estimation. So get used to seeing pie charts instead of bootstrap support values from now on. You can also do population genomics. So this is a membership probability from some of our data in Coral ELISA. And so we use ISSR sequencing, which I'll talk about later. This is a discriminant analysis of principal components. So basically showing clustering based on SNP data. So you can do all kinds of analyses downstream. Okay, so the next part of this talk I want to focus on is on reduced representation methods for sampling the genome. And these are largely powerful with population genomics, phylogeography, but they're also popular methods now for kind of lower level phylogenomics. So the species level, the genus level, etc. Okay. So this is a famous example of sticklebacks and ecological divergence. So looking for patterns of selection in the genome, and also with the development of genotype by sequencing and RADseq, which we'll talk about here in a second. So again, we're back to research questions. What is my research question, and which method will best help me address it? Okay, and so your question will determine how much data you need, or how many data you need across the genome, right? So at the lowest levels of genomic representation, you can build phylogenetic trees and study population structure if you have unlinked loci across the genome. However, if you're interested in doing things like pedigrees or association mapping for traits and population genomic scans for divergence or adaptive evidence of local adaptation, you're going to need deeper and more dense sampling across the genome. And what I mean by more dense sampling is basically you're going to need more SNPs across the genome. So for example, you can catch these different linkage groups. So if you have a locus that's under strong selection, you want to get a SNP that's nearby or even really close by that SNP that's under selection. that's hitchhiking along with it. So the more deeply and densely you sample the genome, the higher probability is you're going to catch these loci that are potentially being selected. Okay, so one of the first methods of reduced representation sequencing was genotyping by sequencing. Okay, so basically what you're going to do is you're going to chew up the genome with a restriction enzyme, then you're going to ligate an adapter and a barcode onto that, amplify it, and sequence it, right? And so you're just going to get like a somewhat random representation of SNPs across the genome from from near these restriction sites. Then along came RADseq, which is actually GBS and RADseq are very similar. They just differ in the number of steps and the orientation of those steps. So with GBS on the right here, you see that you do digestion, ligate your adapters, pool your samples, do a PCR, and sequence them. With RADseq, you do a digestion, ligate your adapters, pool them. You do some shearing, and this is to control the size of the fragments. Then you do size selection. You ligate a second adapter onto that, do a PCR, and then sequence. So that was the main difference between the original representation of RADseq and then GBS, its predecessor. More recently, and a more popular method has come along called ddRAD, that's double digest RAD sequencing. And so instead of just using one restriction enzyme, you use two. And typically you use a rare cutting enzyme, so one that cuts at fewer places across the genome, with a more common cutting enzyme, which has actually made things a lot cheaper. So the way this works is you digest with a rare and common cutting enzyme, and then you do size selection. So you get a really tight distribution of fragments. Then you ligate your adapters, pool your samples, amplify everything, and sequence it. So that's ddRADseq. So there are some advantages and sort of challenges or roadblocks to RAD or GPS approaches. So it's really relatively inexpensive per sample. You get huge amounts of SNP data, up to like 10 to the 7th SNPs per data set. Useful for population genomics all the way to, say, like pedigree analysis, depending on how much of the genome you're sampling. And it's a large driver of non-model genomics. Criticisms, it doesn't sample enough of the genome, so a typical protocol wouldn't sample enough of the genome for robust estimation of adaptive polymorphism. So you really need a lot of genome sequence. So you might want to use multiple restriction enzymes across the genome. The downside there is you need a lot more sequence coverage because you're sampling more of the genome. And if we go back to our coverage formula from earlier, that means you're going to need more sequencing, so it's going to get more expensive. It's not going to be cheap anymore to do this. And so some people say, well, why not just resequence whole genomes? Well, if you work on an organism with a very large genome, that can be prohibitive. Okay, so there's a need for high molecular weight intact DNA for RADseq or GBS to work, and you need a relatively large amount of it per sample. That's getting better as the methods improve, but still you do need, so you're pretty much limited to fresh samples here, so contemporary samples, although people have used RADseq with some success with, say, herbarium samples. But you're not going to get the same amount of information you get from fresh samples. Okay, so every sample needs to have a successful digestion step, whether you're using contemporary or historical samples. So degraded or low yield samples, herbarium samples, ancient DNA especially, if you work in the tropics and collect under hot, humid conditions, you know that your samples your tissues start to degrade rapidly, and so you need to preserve them, and it's really difficult. One of the solutions to this problem is to use RAD or GBS or some other reduced representation library method to build your own capture probes. And that's become really a hot topic in the last, say, 5 years. Okay, so again, you want to consider your research question hypotheses before you start a study like this. Am I studying phylogenetic relationships, population structure? Do I need deep and broad sampling of the genome for genome scans, for association mapping, etc.? How much of the genome do I need to sample? What's the taxonomic breadth of my samples, species level versus population level? What's the genome size of my organism? Am I working with a huge genome, a tiny genome, whatever? What's the repeat content of the genome? Ploidy, is it a model, non-model? Do I have resources I can use, for example, to help me do something like in silico digestion? So So I can use a computer program to scan across the genome and find every cut site for a certain enzyme, and that can give you an idea of how many fragments you're going to wind up with and what representation you're going to get across the genome. So that can be a really powerful thing for you, say if you have a closely related genome that you can use for planning your project, if you're going to use a restriction-based method. The logistics, what's the lowest cost per sample or per SNP that will allow me to adequately answer my research questions hypotheses? So that's always the thing I'm concerned with. So like what's going to give me the best bang for the buck, right? Okay, but when it comes to reduced representation, we've talked about two of those methods, GBS and RADseq, but in reality, there are like many different methods, and it's kind of mind-boggling. And we're also going to look at this paper by Campbell et al. From 2018. Okay, so there's the original RADseq from 2008, GBS from 2011, although various precursors had been around since like 2000, right? And so there are different paths you can take here. There's DDRAD and all the derivatives of that. And one of the points of this paper is like, can we please stop naming these methods? There are just too many of them. But I'm going to focus on a couple over here, like HiRAD and HiRAD-X and RADCap, for example. These are all methods by which you actually create your own probes in the lab from RAD sequencing or from RAD digestions, and then use those for sequence capture. Okay, so the first is HiRAD, and I think a lot of people on the EPSCoR project are going to be using HiRAD or some derivative of it. So basically, you generate RAD probes with biotinylated probes that can then bind to magnetic beads. You do a sequence capture based on the probes you created, and then you get pretty good representation across the genome. And these methods have the added bonus that, like, one of the things you deal with say like RAD sequencing with GBS is restriction site dropout. If a restriction site has a mutation or a substitution that keeps that site from being cut, then that's going to look like an allelic dropout for that sample. If you use a RAD approach where you use one or a few fresh samples to create your RAD libraries, you're still going to be able to capture that region even if there was a mutation that affects the cut site, because there's going to be enough sequence similarity around that to bind to your probes. So it actually reduces the problem of allelic dropout if you're using a sequence capture based methods with RAD probes. HiRAD-X, in this case, where you're using RNA sequence or RNA extractions and then cDNA library building, and then you biotinylate these RNA probes, which tends to increase your capture efficiency. So this is a paper from 2017, the year after HiRAD was produced by largely a bunch of the same authors. EEC is a similar one to HiRAD-X, but basically you're creating your own set of exome probes. So you're using expressed RNA-seq, and messenger RNA to build your probes, and then doing a capture that way. So if you're interested in capturing whole exomes, or at least the expressed exome in a certain tissue type or multiple tissue types, that could be a really powerful approach. One that we've tried more recently, because it's pretty easy, is AFLP-cap. It's very similar to like a RAD approach, but in this case, you're using a PCR to do selective amplification. In that case, you can do a PCR with a 5' biotinylated primer or oligo, and that sticks a biotin molecule onto your probes, and then you can bind that to your samples, capture it with the beads, and then sequence what you capture. So we've had some success with this. As opposed to restriction-based methods, there are also amplicon-based methods. And so a lot of people have been hesitant to do amplicon-based methods because if you say have a genome or a transcriptome, you'd often have to design thousands or hundreds of primers to amplify the regions of interest, and that has all kinds of issues, and it's expensive upfront to buy all those primers, and it's usually only specific to one project. So a couple of other methods have come out kind of using more like of random amplicons. So in 2015 there was MIGSeq, which uses tailed primers, tailed ISSR primers in multiplex. So ISSR based on our other talk. These are going to be basically like microsatellite motifs. Okay, so like CA, CA, CA. N with a tail on it. So you do a first round ISSR amplification and then a second round PCR. And in one of those, the primer includes a barcode. And so basically you'd be doing this the same way you do like fungal or bacterial amplicon sequencing across the genome. You get a few hundred to a few thousand SNPs with this method. I'm not really sure why, but that's useful in some sense, like for basic population genetics, it can be useful. But for deeper scans of the genome, probably not. This is relatively inexpensive and pretty easy. It doesn't require actual library preps. We were also developing in parallel a similar method called ISSR-seq. It's not published yet, but it's in review. It's available on bioRxiv if you're interested. So our approach is just to use single primer ISSR amplicons, pool those across many primer reactions, so you get like lots and lots and lots of bands together, and then we put those amplicon pools through a standard set of a standard library prep protocol. And in this case, we're actually sequencing the whole fragments. So instead of just sequencing like little fragment ends and then size selecting like they do, size selecting and then sequencing fragment ends like they do with MIG-seq, we're actually sequencing the whole fragments, and we get a lot of variation on, in comparison to say something like RAD-seq. So we're getting like 46,000 SNPs in an endangered genetically depauperate species. This is Coralorhiza bentley here, known from about 10 populations here in Eastern USA. So we get a lot of really useful information out of this. So keep your eyes open for more on that. Okay, so analyzing reduced representation data. So there are some, again, dedicated pipelines for analyzing GBS and restriction-based methods like RAD-seq. So STAX is one of them that's really popular, and now STAX2. IPyRAD is another one. So this is a Python-based implementation that allows you to basically filter and assemble your different RAD loci, for example. There's one called ddocent, which I'm not really super familiar with, or you could build your own pipeline. And that's kind of what we did for ISSR-seq. We did more of a standard like mapping reads and calling SNPs and putting them through all kinds of filters. So you could build your own pipeline if you wanted to. Okay, so I'm going to wrap it up there. Hopefully this was informative, and let me know if you have any questions. Thanks.